Hello, everyone. Thanks for your interest in this presentation. The main message of this talk is actually spoiled right here in the title. But let's see in the next few minutes, what does this mean and why do we think this is important? Given the growing human population and its impact on ecosystems, management and conservation practices are faced with increasing frequencies and magnitudes of various disturbances that occur on different scales. Aspects like climate change, terrestrial biosphere degradation, or sea level rise are all well documented. And indeed, disturbances related to human activities like temperature rise, pollution, or wildland conversion are all increasing and expected to continue to intensify in this and the upcoming century. This poses a threat to global ecosystems and to humanity itself, with some experts stating that current trends would push Earth into the 6th mass extinction event. So hence, more than ever, we need to study disturbance, which is important in ecology because it is considered a major factor influencing biodiversity. It is an event in time that disrupts the structure of a community by changing resources, substrate availability, or the physical environment. And while a disturbance might result in inhibition, injury, or death for some individuals in a community, it also creates opportunities for other individuals to grow or reproduce. Succession, which is the sequential replacement of a species like in this image, often occurs after disturbance. And moreover, disturbance can radically alter trajectories of ecosystem dynamics, leading to alternative stable states that may or may not be reversible. So to do this, we use microbes, which typically exist as diverse, complex, and dynamic communities and are involved in all biogeochemical cycles. Water-engineered systems depend on complex microbial communities of living organisms that interact with themselves and with the environment. We can control these systems and measure their functions more easily than in traditional field studies. Also, most bacteria in activated sludge have generation times of less than 24 hours. So hence, study periods of just a few weeks can represent around tens to hundreds of generations of many different microbial taxa, allowing for a faster detection of relevant ecological patterns due to disturbance. But for that, we also need frameworks. And here, ecological theory can help us draw generalized conclusions from specific observations of organisms in their environment by allowing us to classify, interpret, and predict the world around us. And this can also be useful for engineers like me who want to manage these microbial communities. So at CELSI, we work with two types of environmentally relevant disturbances using activated sludge reactors: 3-chloroaniline, which is a xenobiotic compound, and organic loading shocks in the form of double chemical oxygen demand addition. We did experiments at the microcosm scale with replicated sequencing batch reactors subjected to different frequencies of disturbance, either pulses or continuous, for a period of 5 to 6 weeks. But then we further experimented at a 2 orders of magnitude bigger scale of volume and longer periods of operations, around 5 to 6 months. And the reason why we did this is because disturbance is multidimensional. So understanding its effects is tricky, as it can vary in type, intensity, frequency, and its effects can also be scale dependent. So, all right, let's now focus on these two studies marked here on the screen. What do you think happens when activated sludge reactors are subjected to different frequencies of disturbance? Well, a concept of ecology that can be used to explore possible outcomes is the intermediate disturbance hypothesis, or IDH for short, which predicts a peak of diversity at intermediate levels of disturbance due to competition colonization trade-offs faced by organisms. However, the IDH explains only species richness, which is just one aspect of the overall structure of the community. This leaves gaps related to other aspects of biodiversity and also the role of assembly mechanisms and the ultimate impact, if any, on ecosystem function. So we hypothesized about the role of stochastic and deterministic assembly mechanisms as shapers of community structure based on observations using different frequencies of 3-chloroaniline addition as disturbances in bioreactors. We proposed that when intermediate disturbance regimes result in less predictable environments, then the stochastic equalization of competitive advantages is what leads to the observed higher alpha diversity. But on the contrary, extreme ends of the range where conditions are recurrent and therefore more predictable, would select for adapted organisms whose dominance would result in a lower alpha diversity. But again, disturbance is multidimensional, and that was only one study. So we tested this new hypothesis with a different type of disturbance, double organic loading, 5 replicates instead of 3 for more statistical power, and for a total of 42 days. So this alteration represents a disturbance for microbes in activated sludge systems due to changes in competition for oxygen, substrate, and biofilm space. The experimental design employed microbial sludge from a full-scale facility, which was used to inoculate 30 microcosm reactors of 25 milliliters of working volume. These reactors were operated in one-day batch cycles where complex synthetic wastewater was fed daily to mimic the complexity of real wastewater, while avoiding normal chemical and microbial composition fluctuations to better control the factors influencing our observations in this study. Results from 16S ribosomal RNA amplicon sequencing showed that after an initial decrease in diversity from that of the full-scale plant inoculum, we found again a parabolic pattern of alpha diversity, which validated the intermediate disturbance hypothesis. Yet further, higher alpha diversity at intermediate frequencies of disturbance was observed for both taxonomic and phylogenetic indices, both unweight and abundance weight. And this is relevant because the IDH was only defined for richness. But moreover, when quantifying assembly mechanisms using the nearest taxon index null modeling approach, we found a significantly higher contribution of stochastic assembly mechanisms at intermediate levels of disturbance, supporting now the intermediate stochasticity hypothesis. There was also a significant positive correlation between the relative strength of stochasticity and all the alpha diversity metrics from the prior slide. But yet more interesting was that the parabolic pattern in stochasticity preceded that of alpha diversity in time. Temporal dynamics of beta diversity showed that communities diverged over time across the range of disturbance frequencies, forming 3 significantly different clusters at undisturbed, press-disturbed, and intermediately disturbed levels. Misclassification errors of canonical analysis of principal coordinates showed that replicate reactors were less similar to each other at intermediate levels of disturbance, while replicates at low and high disturbance frequencies were more similar. Likewise, community assembly assessed via the beta nearest taxon index showed a higher relative contribution of stochasticity at intermediate levels of disturbance, with beta NTA values closer to 0, indicating that phylogenetic turnover across within treatment replicates was closer to the null expectation. General functions of carbon removal and sludge settlability improved over time of the study, and similarly, the overall strength of deterministic processes increased with time. Hence, correlations between these functions and NTI were positive and significant, indicating improved performance of these particular functions under stronger deterministic assembly mechanisms. Further, carbon removal was better when alpha diversity was lower. So then, a more diverse community does not necessarily translate into better ecosystem functions in this case. On the other hand, nitrogen removal via nitrification was positively correlated with stochasticity, being higher at intermediate disturbance frequencies during the initial stages of the study, where stochastic processes were more prevalent, as we saw in earlier slides. Such correlation, however, disappeared at later stages of the study. So then, why are all these findings exciting? Well, they show for microbial systems that changes in community assembly precede changes in community structure. And plus, stochastic assembly operating at intermediate levels of disturbance could be the reason why higher diversity does not necessarily mean better function. We believe that the ISH framework has actual potential to anticipate changes in biodiversity on an increasingly disturbed planet by directing our focus towards disturbance-induced changes in community assembly mechanisms. So then the framework could be applied to identify cases when disturbance-induced stochastic assembly promotes alternative stable states of community structure that compromise or enhance ecosystem function, so as to design mitigation or intensification strategies. It can also help to develop functionally resilient communities that do not occur naturally via the stochastic mechanisms that are triggered at intermediate frequencies of disturbance. And it can also help toward a general understanding of disturbance-induced changes in community structure and function by integrating the influence of the underlying assembly processes. But again, disturbance is multidimensional. Hence, for this, we call for more research on different microbial systems, types of disturbance, and scales. But if you're curious about this topic, this publication describes in detail the work I have just shown. And I would also like to invite you to check out some of other aspects of ecological theory for disturbance that we have explored at CELSI using activated/reactors. You will find the links to these papers in the description of this video. But to conclude, I would like to acknowledge Professor Stefan Wertz, as well as many others who have also provided support for the work I referred to during this talk. And thanks a lot for your time watching this video, and please contact me if you have any questions.